Right, welcome back. Another episode for the afternoon can be recorded, and this one's going to be looking at the checkpoints in the cell cycle. If you remember the last lesson that I popped up, I specifically mentioned where the checkpoints were, but we didn't go into too much detail about what they checked for, or even what happens when they find something that shouldn't really be there in the cell cycle process after mitosis or in any of the stages. So let's talk about that today. Alright, so here we are. We know that mitosis, the cell cycle, the whole purpose of it is to go from having one cell to having multiple cells that are exactly the same as the parent cell that they came from. So we call these cells that come off or that are made from the parent cell, we call them daughter cells. Now, sometimes the process isn't perfect, and we do in fact end up with a cell that has more DNA than it should, and that can be a bit of a problem. Also, we can end up with cells that have less DNA than they should, and we also should know that that is a problem. Also, we could end up with cells that have DNA that is not good, mutations in the DNA strands, which that can also cause a lot of issues. So how does the cell cycle try to prevent this? Well, there are checks at each of these stages in the cell cycle. The first one, the G1 checkpoint, it checks for any damage to the DNA before it goes into the synthesis phase. It also checks that the cell has efficient nutrients and that it has gone through enough size and growth to actually survive the rest of the stages. If it hasn't gone through enough growth, if it doesn't have enough nutrients, or if there's any DNA damage, then the checkpoint halts the cell here. At the end of the G2 stage, there was also another checkpoint, and this one actually just checks to make sure that the DNA that has been replicated has not been damaged in the process, or that there is any errors after the S phase. Sometimes after replication, you may have ended up with too much DNA, not enough DNA, or DNA that has been had some other type of mutation that has occurred. It's important that we try to make that the cell is checked after these stages to make sure that the mutations are not harmful, or to make sure that the DNA that has been checked is actually all there. The third and final stage was the checkpoint was the one between the metaphase and anaphase stages of mitosis. It checks for proper spindle fiber formation and connection and attachment to chromosomes, So they're the two things that are checked for at that stage. Because before that, prior to that, there should have already been enough of everything already checked in the prior stages. There should have been enough nutrients in the cell, and there should have been no damaged DNA. Now it's just time to make sure that the spindle fibers have in fact connected, and that that they're formed properly as well. Now, if at any stage in any of these checkpoints it turns out that the requirements were not met, then CDK, or cyclin-dependent kinases, stop the cell cycle process for that cell. So stop the cell cycle and initiate a pause button. And from there, it's time to see what we can do about repairing the damage done or repairing the cell. Now, for some of them, it can be as easy as a fix as let the cell grow more, let the cell get more nutrients, or try and form those spindle fibers. But for the specific examples where DNA is the issue, the stuff that is stored inside the nucleus that offers the blueprints for how cells function and how proteins are made and how everything is done, well, then there is a very specific process that needs to be adhered to. So CDK, cyclin-dependent kinase, it's an enzyme. It pauses the cell cycle. What it does is it activates a gene inside of the cells called the p53 gene. A gene is a segment of DNA that codes for a specific function, and in this case, it codes for DNA repair. So DNA actually has its own coding to say, hey, we're going to try and repair ourselves. Option 1 is quite simple. P53 establishes that the DNA can be repaired. It activates a cascade of enzymes that come along and try to perform the repairs. And once the cell is back in action, the cell cycle continues. CDK deactivates and cell cycle continues and replication can continue. Option 2, not so great. Option 2, the DNA is deemed irreparable, and apoptosis is initiated. What's apoptosis? Apoptosis is programmed cell death. I'm going to be very specific here. If you are answering this question in an exam, whether it is year 12 or year 11, you need to be very, very clear that apoptosis is programmed cell death. Cell death is not correct because cell death can also be necrosis. Necrosis is when the cell bubbles and boils and everything dies inside of it without it being properly disposed of, and that is bad. Necrosis is bad. Apoptosis isn't necessarily bad, okay? You can also not say something creative. Pardon the trigger warning from this video as well, my apologies, but you can't write something such as in terms that is like cell suicide, because that's not true. The cell does not do that. The cell has a programmed death, which can be initiated from outside of the cell or initiated inside of the cell. And either way, we're going to look at this where the DNA was unable to be repaired. P53 has activated the apoptosis pathway, and the first thing that happens is that the DNA and nucleus are dissolved. The DNA is chopped up by a particular enzyme, which then goes ahead and chops up the rest of the DNA from around the cells, goes up and chops up, ribosomes, it chops up tRNAs, it chops up all these other enzymes inside of the cell and proteins inside of the cell, deconstructing the cell. In the process of deconstructing the cell and all of its organelles, it also deconstructs the cytoskeleton, giving the cell its shape. The cell then goes and starts to bead. It has bulges that start to appear, and these bulges are then filled with the cellular contents, which has been chopped up into tiny, tiny little pieces. So this is broken up bits of tiny the broken tiny bits of cell are inside of these bulges, which then float off and start to disintegrate. They become one of my favorite words to say in biology, bleb. They're tiny little blebs filled with cellular contents. What happens next is that a white blood cell, a macrophage, will come along, consume that bleb, break it down with the lysosomes inside of it so that the cellular contents is not harmful. It won't go ahead and create a toxic reaction inside of the bloodstreams or cascade and toxify other cells, which is different to necrosis. Necrosis will break apart, will spill cellular contents into the surrounding area, which will attract macrophages to eat that, but then other cells will then get damaged in the process. It's when you end up with those really, really bad sores. So that's apoptosis. It's programmed cell death. It's not cell suicide, and it's not necrosis, which is just plain old cell death. Okay? Really important that it is programmed. Now, if we're looking at this image here, why is it important to have apoptosis? Well, the first example that I can show you is that it's important to have apoptosis for embryo development, for the development, from us going from being infants into adults. Our toes and fingers were all joined together by webs when we're little tiny embryos, and those webbings need to break apart, and they do that via apoptosis. The cells that make up that little webbing dies. It goes through programmed cell death and disintegrates, disappears. The other reason, other than mutations, is when our cells become infected from an outside source. We want those cells to not proliferate, they send out their own warning to some other, to other cells and say, hey, I'm damaged. And so programmed cell death can happen to cells that are infected with viruses. Now, what's the consequence if we don't have apoptosis? This is where some of the malfunctions come into it. Not necessarily all of them negative. The first one for the embryo development is that we can end up with some pretty interesting physical mutations. This one here is called syndactyly. It's where your toes and fingers, the webbing never disappears. And you can end up with joint toes and joint fingers. This is actually my foot. I have syndactyly of my toes. Please don't judge me. I understand that they are not great feet. Obviously, when it comes to cells that are infected with viruses, the virus spreads and it continues to proliferate, and we can become very sick with viruses still present in our bodies. But the one that we really, really need to focus in on is what happens when we have a mutated cell whose mutation is present in one of the two genes that I'm about to mention. Those two genes are proto-oncogenes or tumor suppressor genes. A proto-oncogene is like the accelerator on the car. It'll speed up the process of cellular replication division. A tumor suppressor gene is like the brakes. It slows it down. Now, if you have a mutation in either of those genes that goes uncorrected, well, we end up with cells that start to divide and multiply rapidly and quite violently. For anyone who doesn't know what this one is commonly called, this is our common cellular behaviors of cancers. Okay, cells that divide uncontrollably without haste, or and then they create large tumors and swellings, and they can make they can hinder our immune systems, and that goes ahead and makes us very sick. So cancerous cells are cells that divide and divide without any kind of halt. Yeah, there's the uncontrollable division. So in terms of disruptions to the cell cycle, it's important that we have these checkpoints to make sure that we check for any cellular damage when it comes into it. Apoptosis is programmed cell death, and without it, we can end up having some interesting complications, such as joint toes and joint fingers, or some serious ones such as viruses and cancers. Hopefully this has been informative. Hopefully you've gotten something out of this lesson. You understand something new, and I'll see you in the next one.